Okay, good morning, everyone. Thank you for coming. Welcome to this seminar of the Cycle Visibles CNB. As you know, this is a cycle that the Equality Commission has initiated last year. And we try to invite women who have been successful in many different fields in life, not only in science. And today we are very lucky to have here Eva Nogales. Eva Nogales is Howard Hughes Medical Institute investigator, a professor of biochemistry and molecular biology at the University of California in Berkeley, and a senior faculty scientist at the Lawrence Berkeley National Laboratory. But actually, Eva studied at this university, at the Universidad Autónoma de Madrid. She did here her degree in physics, and then she moved to the UK to carry out her PhD in the Daresbury synchrotron radiation source, where she started studying tubulin, the structural dynamics of tubulin. Then she moved as a postdoc to the Lawrence Berkeley National Laboratory in California, and there she produced one of the first high-resolution structures in cryo-EM using electron crystallography to show the organization of tubulin. She then started her own group, and since then they have very successfully applied and developed cryo-electron microscopy to very important and very challenging, often biological complexes. For example, they have worked on the machinery that controls gene expression, and also they have kept working on microtubules and tubulin. She has over 200 publications, and these publications have been cited over 30,000 times. She has, as you see, she has carried out all her career far from Spain, but she has kept connected to the Spanish science, for which we thank her. And today she's going to tell us about some of their latest results, but also she will take a couple of minutes to give us her vision of working as a woman in a field in biology where there's still a considerably larger gap between the different genders. So thank you, Eva, for coming, and the floor is yours.
Thank you, Carmen. There was a tradition of coming here to the National Biotechnology Center on an annual basis, and we broke it for obvious reasons. So I'm very, very glad to make it back. And I'm going to try to say a few things, just maybe for discussion. We can take it as part also of the questions at the end concerning the issue of women scientists, women in science, and one particular aspect that I decided to emphasize. I want to give you a background, and I will be talking about how much or what was the lack of female reference that I suffered myself as I went through my career. I think things have changed, but maybe not that much. I'm very local. I'm from Colmenar Viejo. And at the time when I was growing up there was only one instituto, one high school, and it was from Colmenar and all the towns around the Sierra de Madrid, and some people from Fuencarral and things like that. And there I had great women reference, because my math, physics, and biology teachers, they were all women, and they were amazing women who I have kept in contact with, and they have now retired, but they remain super active. One is writing physics textbooks. The other one is, what's happening? Oh, okay. The other one is still helping students with writing the science report newsletter that they have, so super active people. But in any case, then I came here to do my undergraduate studies, and this, sorry, this is a picture that I found that correspond to when the Autónoma was started, and that was not maybe 10 years before I came here. So it was a relatively new university then. And I studied in the physics department. There wasn't a single female faculty scientist. So there was no reference there for us whatsoever. And the whole of the department had one female graduate student, which I believe is now a professor in that department. So from there, I moved to Dasbury, and my PhD supervisor was Joan Bordes. And there at Dasbury, so this is a synchrotron source, is closed down now. The synchrotron is now down in Oxford, so things have moved politically down once again from the north of England. That's what the synchrotron was. This is the Van der Graaf accelerator that was closed after Margaret Thatcher visited the lab. It was like the touch of death. The synchrotron kept going. I actually talked to her, and that's where I found out that she had a PhD in chemistry and had done crystallography, actually, of small molecules. There were some women there, but not in positions of power. And, you know, so there were several of us graduate students working there, and there were a few staff scientists. So from there, I moved across. So this is something that, you know, I'm using this to point out that I think is really good for people. You know, it's a good thing that scientists move around. You don't have to leave your country and do what I did, which is to live somewhere else. But I think it's really enriching for many reasons, not just scientifically, but also in the way you look at science, and to give you a perspective that you have the opportunity to spend time abroad. It could be a short period doing your PhD, a short postdoc, it could be going on sabbatical, whatever. But I think it's really enriching. It is enriching for me to spend time in other places and to see how science is done, even if you then come back to Spain and you have that kind of background to be able to then, I don't know, influence how things are run in your own institutes, or how you communicate with people maybe of power that can change how things work, including how it works for women, by the way. Okay, so I moved to the Lawrence Berkeley National Lab. So this is yes, another circular building, means another synchrotron. We went there because my boyfriend then, now my husband and father of my two children, was hired to actually build all the beamlines in the synchrotron. And I found a position working with Ken Downing. Unfortunately, Ken died the summer of 2018, but he was an amazing mentor. He was one of the pioneers of electron crystallography. And here it is. This was our team: Sharon Wolf, who is now works at Israel, Ken and myself. And this was the team that worked on the structure of tubulin. That was a postdoc that was very risky. And I knew it was going to make me or break me. It worked out. That's why I'm talking to you today. Otherwise, I don't know what I would be doing. So I was able to get a position in the molecular and cell biology department at UC Berkeley. Now, UC Berkeley is an amazing place. I was super lucky. Just the intellectual, it's an intellectual powerhouse. And the colleagues that I have there, the people that I have been able to rub shoulders with, have really influenced what I've been able to do in my career. Just to give you an example, well, I was super lucky also because I was at the right time at the right place to get Howard Hughes, and I've been a Howard Hughes investigator now for 22 years. So that has made a big difference. Just an example of the people that I rubbed shoulder with. This is Jennifer Doudna and myself. This is a picture that they took for one of our early collaborations, maybe on the on translation. We worked then in siRNA, and then we've been working for many years now on CRISPR. And I want to mention Jennifer because she is an amazing role model. She is like, you know, there are many Nobel Prizes. I know many, and some of them are, you know, like to a penny, honestly, but she's not. She is special. She is truly special. She got the Nobel Prize for CRISPR, but she could have got it for many other things. And she has never slowed down. She started an institute. She has 5 or 6 companies. And she runs a lab, directly runs a lab, is involved with the science of, you know, something like 25 people. she's almost non-human, and she's gorgeous. She looks like a model. I don't know what to say. In any case, she is also modest, appreciative. She's a dream come true, and I think partly she's not someone where the fame has gone up to her head, and I think that's partly because she's a woman. But that's an idea. All right. So this brings me to one of the points that I want to bring in the context of science, women in science. So there could have been many issues, but I thought I would dedicate a few minutes to the issue of recognition of women in science. And I just chose the top recognition award in science, which is the Nobel Prize. So just some data for you that you may or may not know. Of all the Nobel Prizes to date, only 3% have gone to women. Okay, so that's 60 women since this started more than 120 years ago. Okay, the thing that is very interesting is, so there is a positive side of things. Things are evolving fast. So this is up to 2021. There's been 2 more women this year, the Nobel Prize in Literature, and one of the Nobel Prize in Chemistry, actually Caroline Bertozzi, who used to be my colleague at UC Berkeley, and actually the work was done at Berkeley that she got the Nobel Prize for, and actually was my neighbor, lived right around the corner until she moved to Palo Alto and Stanford. In any case, things are getting better, but this is still very bad, and I just have to show you this. So of those 60 women, this is how they distribute in percentage of what Nobel Prize they got. And I want you to realize that, 30% of them was the Peace Prize, about, so a third Peace Prize, about a third literature. And then this is what has gone to science and economy. Is like they don't exist, really. This is very different from the distribution of men. So there is hundreds of men that got the Nobel Prize, of course. And see how it's dominated. Almost 3/4 of the Nobel Prizes are actually in the sciences, in physics, medicine, and chemistry. So women are making it, but of these, very few are actually scientists. So what are the reasons for this? So there could be many. You can say, oh, just, there's fewer women, or they're not as good as scientists. And, I think there are other things that are precluding women from reaching those heights or being recognized, or a mixture of the two. So these are just 3 that we could comment afterwards if you want. I think nature is against us. Biology is against us. Why? Because our biological clock and the academic clock are highly incompatible. Just think, when is the time for women to be having children? When we're either doing our PhD, doing our postdoc that will give us the breakthrough to be able to apply for a position, or in the process of getting tenure at least in the American system. And because of how we see the process still in our society of women being in charge, the main people in charge of the children, this is affecting women having children more than men having children. It is very rare that a man goes and says, well, this is going to be very difficult, and maybe I have to decide between having a family and having a scientific career. I don't know of any man that has ever had to have that kind of question in their mind. Okay, I think society, mostly men, but actually also women, is against us because women are still overlooked and unrecognized. Honestly, there are certain things that I know women have done that if they had been a man, they already would have some big prize. So why this happens, I don't know. I think it's because of biases that we still have in our mind of how much the work of women is appreciated or not. And then there is this one. We women are sometimes work against ourselves. And I'll give you one example. Women do not self-promote the same as men. This is not my opinion. This is not anecdotal. This is data. People have done, sociologists has done work about this. The Howard Hughes, some years ago, decided to go back through a different way of getting candidates. that became self-nomination. So instead of going through a number of institutions, say, UC Berkeley, you have to nominate 5 people or 3 people this year. They just say, we're going to do it open because we want to be more democratic. And this is going to be by self-nomination. The number of people, of candidates that they were being considered being women plummeted because women just have these self-doubts. Oh, I'm not good enough. I see it in my own lab. I see graduate students, postdocs, male and females, and the men are boasting for whatever thing they do. And women are, yeah, but they could be done better. And I don't know if I'm good enough. What the hell? Why is this? Is it hormonal? I don't know. But I talk to other women, including women that are really, really successful. And this is an issue. And I don't know what to tell you. Remedies. So there are remedies that can be institutionally set. So obviously, this can be helped by facilitating, not making women having to make a decision or a compromise when it comes to having a career and having a family. I think, unfortunately, in the sciences, I don't know what you think, but I think science is such a race that it's not like you can stop your career for 2 or 3 years and then come back easily, at least as an independent competitive PI. It's really hard. Maybe someone has done it. I think Carol Robinson did it very, very early in her career, but she's special. That's why is, what is it? Madam, no, what is the equivalent of Sir? Is Dame. Dame. She's a Dame. That's why she's a Dame. But at least, you know, if that is not an option, there are certain things that will facilitate that women, you know, don't consider this to be a trade-off, is one thing or the other. You know, in places like the United States, the academic clock for tenure can, you know, can be moved back forward by a year per child. That helps. Having very, very good childcare next to where you work, maybe in your own institution, will help tremendously if the resources were economic resources were available for it. This is partly, you know, is a problem that has to be that, you know, has to do with unconscious bias. One of the things that, you know, we have to promote and we have to make very visual those women that really succeed, so that the image that someone has of a scientist is not this white man, typically with gray hair like mine. That's the one thing. But that there are other images of scientists, and that we stop thinking that women, for being women, are not gonna, they're not the leaders of the research, they're not the ones having the original ideas. And for this, I don't know, talk to other women, realize that we all have these kind of doubts and get over it. And just, what do you have to lose? Promote yourself, apply for things. The worst thing that can happen is that you don't get it. That's the same as not, you know, better than not try. So that's it. That's what I want to say. And now I'm going to move on to science. If I have time, I don't know if I'm going to make it to the end. So this is a particular technique that actually got the Nobel Prize for 3 men. That's okay. Particularly like this. It is a technique. Is not, the video is not running, but that's okay. So the idea is that these molecules are in solution, they're going around, they're flopping around, they are doing their business, biochemically active, they're placed on an EM grid. We blot it to a thin layer and we very quickly freeze it. And it's this frozen hydrated state that goes into the electron microscope. And then we have to do certain magic. The low temperatures allow you to get in an aqueous solution that is now maintained at liquid nitrogen, cannot sublime in the vacuum of the scope, but also reduces, although it doesn't eliminate radiation damage. So we still collect data, very pixelated data, with low doses. So it is very noisy, but we collect many, many images of these objects like ribosomes that then are processed computationally to classify, to align, to average, improve signal to noise. And then when we combine images like this from many different views, we can obtain a 3-dimensional reconstruction. The whole point is that the crystallization is avoided And these allow you to study fully assembled complexes rather than crystallizable units in conditions where they're biochemically active. Very interestingly, it allows you also to look at different functional states, even when they coexist in the sample, and where the heterogeneity is either compositional or conformational in nature. This is very cool because this is just an example from our own lab where we were looking at human TF2D. This is a factor that is involved in core promoter core sequence recognition of sequences in core promoters for POL2. And it is over a megadaltonic size. It has one domain that is almost half a megadalton that moves 150 angstrom. And in the process, it keeps the TARA binding protein in check until all the certain domains recognize downstream core promoter sequences and only deploy TBP at the right place when a number of checkpoints have been fulfilled. And we could study that process and get structures ultimately to both the APOR structure or the DNA bound structure in this context of mixed state that ultimately make things very difficult, but also carried out a lot of biological information. We could do this in this particular case because this technique requires very small amounts of samples. So it's very common in my lab that we work with as low as a few microliters of things that are in the tens of nanomolar concentration. We concentrate the sample on the grid, but we can deal with this. And why is this very interesting? Because it opens the door, if so you desire, to study endogenously purified complexes, not overexpressed, but endogenously purified complexes that may exist in very few copies, like is the case for TF2D, RNA polymerase 2, TF2H, or, for example, one of our latest transcriptional coactivator complex, SAGA. In this case, for other studies, we have used immunopurification. So we had an antibody and an elutive peptide. In this particular case, instead, we generated a CRISPR HeLa cell line that was tagged in one of the unique subunits to this complex. And then we just pull on that tag and obtain enough material that by concentrating in the grid, we could obtain the structure. So those are all the studies that have to do with transcriptional gene regulation that are based on the classical model that when we think about gene regulation of sequence-dependent transcription factors that recognize DNA sequences and ultimately do something, typically recruit chromatin remodelers, recruit general machinery to ultimately, none of the animations are playing, I don't know, to allow for polymerase to transcribe the gene multiple times. And in this context, nucleosomes actually represent obstacles. What I'm going to tell you about has to do with epigenetic regulation. And this is a regulation that is actually mostly independent of DNA sequence, where the nucleosomes are the substrates for regulatory enzymes. So these epigenetics are based on both readers, writers, and erasers of both DNA and histone modifications that ultimately regulate transcription by defining the compaction of chromatin from an open state that promotes transcription to a compacted state that represses it. So this is the complex that I will be telling you about is polycomb repressive complex 2. This is a complex that is essential for gene silencing during development to define cell identity and also to maintain it in the life of the adult organism. That's why it is mutated often or misregulated in cancer. The complex, which has 4 core subunits, has as a role in life to trimethylate lysine 27 in histone H3. And this subunit, ECH2, is the one that has that catalytic activity. And the complex, interestingly enough, I'm going to be telling you about this a lot today, has a subunit, EED, is a WD40 beta propellers, I'll show you in a second, that actually recognizes that modification and then allosterically activate the complex. And it's thought that this is very important to propagate this modification to entire loci in the genome. The complex doesn't ever exist, we don't think, as those core subunits. Instead, it interacts with a series of protein cofactors that come in different combinations to give rise to alternative PRC2 forms. And these cofactors contribute to the regulation of the complex, most often activating the complex. And then there are, of course, histone modifications that will regulate its activity. I just told you about one, which is the own modification that the complex impart, but the complex can read many other modifications, and some of them are repressive and some of them are activating for PRC2. So the combined effect of cofactors and histone modifications, and there's a very interesting crosstalk, I'll tell you about that in a second, can result in the activation of the complex, which result in the spreading of the modification, the compaction of chromatin, and the silencing of genes, or they can give rise to the inhibition of the complex, which allows for chromatin to remain open in the absence of this modification. So we started working on this complex some years ago, before direct detectors made possible the study of complexes that were smaller than 0.5 megadaltons. This was work by Claudio Ciferri when he was a postdoc in my lab. Now he's the head of structural biology at Genentech across the bay. So this is just the domain representation of the subunit. Again, ECH2 is the one that has the set domain that carry out the methylation. And EED, which is a WD-40 beta propeller, is the subunit that actually does the allosteric regulation. Okay, so Claudio overexpressed this in insect cells, produced a complex, and when he looked at it, it was impossible to get a structure. And this is because the complex was very, very flexible. He decided to add one of the cofactors, ABP2, and he realized that in the presence of ABP2, the complex was stable enough. And he carried out negative stain because at the time, this, which is about 300 kilodalton, was far too small for cryo. He was able to get a structure as good as you can get with negative stain, which was about 20 angstrom. And at that resolution, WD-40 domains, and there are two in the complex, look like donuts. And there was such donuts, two of them in the complex, but we couldn't tell anything else. So because he had a recombinant system, he decided to do something that I think was really cool. I still think so, which was to use labels that could be visualized directly in the images, either MBPs, N and C terminal, or GFPs that he introduced between domains or even between the beta blades of the WD-40 domain. So this is the idea. This is PRC2 by itself, the wild type, sorry. This is the one that is modified with a GFP right there between the first and the second blades. And the appearance of the density, extra density, tells you where that position is in the complex. This is another one where the GFP was put farther down in the sequence, and this has moved. It really is tracking the beta propellers going around this donut. And when he did this systematically through all these domains, he was able to come out with a model of where the different domains within the different subunits are. These were just docking without accuracy, just putting placeholders for all of those predicted domains. And I'm showing you this because it turned out to be pretty much right. So this was the structural information that we had on PRC2 for a number of years. But very interestingly, it turns out that the top lobe of PRC2 is actually the minimal functional subunit that can do methylation that is autoregulated through allosterate. And that's one with the full ECH2, the full EED, and the C-terminal domain of SUS12, this VEVS domain. So this region was then subsequently crystallized, first for a fungi and then for human. And those crystallographic studies, they were very beautiful. They actually worked out the allosteric mechanism. So they worked with an activating peptide, so a piece of H3 that contains a trimethylated lysine 27. And what they saw is how it bound to the beta propeller, shown in cyan, and how, as a result of the binding, this structure, which they call the stimulatory response motif, and I'm going to call SRM from now on, became folded. So in the absence of this peptide, this region is invisible in the crystal structure, is disordered, and upon binding of the peptide, this helix folds on the surface of EED and interacting with the peptide. And in the process, interact with this helix within the set domain and changes the conformation of the active site to be a one that can actually carry out the methylation. So we have to move forward and get the structure of the whole PRC2, and we face incredible odds. And this has to do with this general problem that in some cases you can overcome fairly easily, and in others is a real pain in the neck. And it has to do with the disruption of proteins at the air-water interface during the process of thinning. And this is by far the single most important limitation in cryo-EM. If we solve this in a general way, so every sample that we put on an EM grid gave us protein that is in no way damaged, we will do the whole proteome within my lifetime or even Jose Mari's lifetime. Okay, in all functional states. It's just that every time we try a new sample, we have to find some kind of surfactant, some kind of stabilization, because this air-water interface is very hydrophobic and it has the capability of disrupt complexes and even partially or fully unfold proteins. And PRC2 was a total pain. So Vignesh was ultimately able to move this into cryo. He did it for a complex that is the so-called PRC 2.2, the one that has a full set of co-activators, which include both ABP2 and JARET2. Okay, and to do that, he had to both use a carbon support to absorb the sample so that there was less likelihood that the sample will be diffusing and touching the air-water interface when the sample gets thin, and also slightly cross-linked with BS3. And he was able to get a structure of enough resolution that several structures of enough resolution that he could get a structure now of the full complex. Now, in this case, this is a structure of an activated complex. And this is because JARET2 is itself a substrate for PRC2. And it gets methylated in lysine 116, and in our overexpression system, it gets methylated, and when we get the structure, here it is, we have, it is methylated and bound to the allosteric site in the EED, so it causes the stabilization of the SRM and the right state for the set domain. Now, so this is just a blown up of that region, just concentrating on those three elements, and this is just to show you that the methylated lysine is in the expected hydrophobic cage, giving rise to this activated state. Now, he could produce a JARET2 that starts at 119, so it's missing this methylatable residue, and in that case, he gets a complex that is in an active state. There's nothing bound to the EED, there's no SRM is unfolded, and the set domain is in the wrong conformation. So that's inactive. So one of the things that he was able to see is where the ABP2, which has this stabilizing effect, sits, and he basically staples the top and bottom of the complex. And he also saw that the C terminus of the complex binds to the other WD40 domain, which had previously been shown to bind to the K4 lysine in H3 in an unmethylated state. So this is another example of how one of the cofactors can also mimic, in this case, an unmethylated histone tail. What the meaning of that is, we still don't know. In any case, just a reminder, JARET2 can act as an allosteric activation. That's part of how this cofactor can activate the complex. Now, we wanted to look at the complex bound to its real substrate, which are nucleosomes, and we decided to look at one particular case, which will be the boundary condition, where one nucleosome is already trimethylated and the other one is not. So one could serve in principles and as an allosteric and activator, while the other serves as substrate. And we did this in the absence of JARET2. So this will make sense in the context of PRC2 complexes that don't have JARET2. where the allosteric activation has to come from this nucleosome. And, this is how PRC2 looks in negative state. This is when it's bound to dinucleosome. This was very cool because both nucleosomes are engaged, but the PRC2, this didn't have to necessarily be the case. The problem was that the cross-linking was incompatible with nucleosome binding. This is in retrospect because it basically titrates away lysines that are involved in binding the phosphate backbone of the nucleosomal DNA. We could also not use carbon because then we would just get always this orientation. So Simon, who was the postdoc that did this work, now has his own lab at the University of Cologne, did cryo-EM, and we knew the complex was going to be damaged. So what happens is he gets the top lobe and the nucleosomes, but he loses the bottom lobe. Okay, this is because the region of interaction between the top and bottom lobe is actually a hydrophobic patch. And this is competed away by the air-water interface, unraveling the bottom half. In any case, because the nucleosomes bound to the top, we could go ahead. The resolution was lower. It was about 7-8 angstrom because of flexibility in the interaction of the nucleosome with the complex. But we could interpret it based on existing structures, both for nucleosomes and PRC2. And this is just to show you that we have, coming from the allosteric nucleosome, peptide bound at the allosteric side, SRM folded, and therefore the right structure of the set domain, which is engaging the tail of the substrate nucleosome. We were able to define something that had not been seen before because all previous studies have been done with peptides, how the complex interact with the allosteric nucleosome through these very positively charged elements, binding with both gyres, in this case of the nucleosome. And again, for the part that binds the substrate nucleosome by the set domain, there are these highly conserved regions of positive charges that are interacting with the DNA. And this constitutes the main way in which the complex interact with these regular nucleosomes. Now, there was something very curious, and is that when we use the known structures, there was an extra density, very robust. We can play with the threshold and it still shows that did not correspond to anything. Now, there were regions of PRC2 that neither the crystallographers nor I had been able to see, maybe because they were disordered. So something there, binding in this very critical position, was visible now in the presence of nucleosome. And the asterisk there marks the position of lysine K27, which we can extrapolate from where the K27 is and where we stop seeing the tail in the nucleosome crystal structure. And I'm bringing this up because trimethylation of K36 is one of those modifications that actually inhibits the complex, and we're working on this right now. But I want to tell you about one that is actually an activator of the complex. And this is this ubiquitination, monoubiquitination in H2A. This is imparted by the other polycomb repressive complex, polycomb repressive complex 1. And this is recognized by PRC 2.2. It was known, it was supposed to be JARAT2. And this brings PRC2 to the side, which then trimethylates K27, which serves as the landing pad for another form of PRC2 that actually compacts the chromatin. So there is this interesting feedback loop between the two polycombs. So we wanted to look at those nucleosomes. We wanted to look at them without having them disrupt, under conditions that disrupted the complex. So we implemented a method that had been proposed and tested with ribosome by Bob Glaze and Bonjun Han at the Lawrence Berkeley National Lab. And the idea is to generate a lipid monolayer where some of the lipids are biotinylated and then give it a streptavidin, give it time, and under conditions where the lipids are fluid, this will form a two-dimensional crystal. And then you can ubiquitinate your sample. In our case, we just ubiquitinate the linker DNA in our nucleosomes. This will bind to the streptavidin monolayer. Then you add the PRC2, or you can mix these two and then add it. You incubate with the monolayer. You wash off anything that is not bound. And now when you block, these things are absorbed and they're not going to detach and touch the awards interface, or that would be a lot more rare. The forming two-dimensional crystals is because you have a background of streptavidin, but if you Fourier transform them in the computer, the contribution of the streptavidin come in Bragg reflections that you can computationally mask out, and then Fourier transform back and recover images that where the streptavidin is gone. This worked very beautifully. Vignesh implemented it. I forgot to tell you that now he has his own lab at the University of Colorado Boulder. So, none of the videos are working. Never happened to me before. In any case, he was able to get the structure and get it with the ubiquitinated mononucleosome and identify the region in the complex that were doing that recognition. And he showed that one of the ubiquitins, that are two because of the symmetry, including a region that had biochemically been defined before this ubiquitin interaction motif, which forms a helix that interacts between the ubiquitin and nucleosomal DNA. But then that footprint is extended to include a second helix that interacts with the classic acidic patch on the top of the nucleosome. And then the other one was actually being recognized by AVP2 using the zinc binding regions in the N-terminus, again, forming a domain that can recognize both the ubiquitin and interact with the surface of the nucleus. The resolution now was good enough that we can see the details of the tail as is being engaged and taken all the way into the active site. This is the K27. Identified all the residues that are involved in channeling the tail. And very interestingly, I'm showing you this because many of the cancer-related mutations go there. So these are mutations that impair the engagement of the tail and render PRC2 incapable of binding. But something that was very interesting is now at this high resolution in this well-preserved complex bound to nucleosome, we could see what this was. This is the extra density that have never been seen before. We call this the bridge helix because it binds between the set domain, the nucleosomal DNA, and the histone tail. So very, very functionally critical element. And this is something that is only folded. This is a recurrent theme in PRC2. It is unfolded and only folds against the nucleosomal DNA. All right. Why am I emphasizing this? Because around the time when we were publishing this, there were two studies from Tom Check and Danny Reinberg, who are big people in the PRC2 community, that shows where ECH2 actually automethylates itself. And it turns out that those are lysines that are at one end of that bridge helix. Now, they showed that the automethylation occurs in cis. So, and you can see how the position of this helix would allow this. So, when there's no nucleosome, this helix unravels, unfolds, and this is the end. This is the end where the lysines are, and this is the active site. So, it will very easily reach the active site and get methylated. Now, they proposed that the regulation, which was an activation of the methyl transbase activity, likely occur in cis. And their idea was that this peptide will be getting into that active site and block it, but if methylation occurs, the complex has very low affinity for the product and it will release it. And now this is in a situation where it can take the tail of a nucleosome. Now, because of where this is, we propose, but we haven't proven yet, that this methylated state could facilitate folding of the helix and or substrate engagement, meaning facilitate the binding of the nucleosomal DNA of the tail. But what we wanted to do, and by the way, when we purify our complexes, these lysines are methylating in our sample. So we already have pre-methylated PRC2, ECH2. We decided to see what would happen if we give nucleosomes to a PRC2 where there is no activating peptide, no JARAT2, no peptide from H3 that is methylated. What will happen? what state would the complex be in, okay? So we just give it mononucleosomes that serve as a substrate, and there's nothing to that is methylated in there that could regulate it except PRC2 itself. So this is work that started by Simon and then Paul Sauer, a postdoc in the lab, continued. There's a lot of flexibility here, so the resolution is not very high, but again, we could interpret it. Now we've improved it. I just haven't had a chance to change the slides, but nothing had changed in terms of the interpretation. Okay, so this is PRC2 is engaging the nucleosome through the set domain like before, but the animation is not going to run. But there is a second PRC2, okay? So this is the one that I was showing you that is engaging the nucleosome as normal. There is a second one that is binding to the first and is interacting with the linker DNA. So we referred to this one engaging with the nucleosome as the proximal PRC2, and this one that is distal that is a distance away as the distal. And again, these interactions, they're all they involve flexible elements of flexible DNA linkers. So the resolution is limited, and we interpret it by docking structures. So just very quickly, because we're running out of time. In fact, I'm not going to do this. I'm going to concentrate on the main point. Okay. I want to concentrate on the key interaction that is established here between, so these two PRC2s interact in the context of chromatin, okay? They interact without the nucleosome, we don't see this interaction. They interact when this is engaged normally to the nucleosome, and this one can, through avidity for the two linker DNAs, can engage in this interaction. What is that interaction? This is an interaction that brings together the set domain of the distal PRC2 to the allosteric ED site in the proximal, the one that is engaging the histone tail. And this is just docking our structure, and I hope you can see that these helix that have the trimethylated lysines, there's no density for it. That's not surprising, because this only folds when it's bound to the nucleosome. It is folded there, but it's not folded here. But I hope that you also see that there is plenty of density where this, in an structured state, could go. And indeed, if you look, there is both density bound to the ED and density corresponding to the SRN. This is an activated complex. There's no JARET2, there's no methylated nucleosome, but this one is providing the mechanism to activate through the classical allosteric site. And if you were to look at this other one, of course, there is an empty ED and there's no density for the SRN. So we propose that in addition to these possible mechanisms for activation in cis, there is for sure an activation that occurs in trans that uses this automethylated ECH2 via the ED to allosterically activate in what is now a kind of universal mechanism for PRC2. Okay, where one of the PRC2 is activated while the other one is using an allosteric effect. We think that this will be relevant in the context of activating the complex in a new site of activity to initiate this trimethylation. So there's no previous nucleosome trimethylated that I can activate, and in the absence of cofactors that have this capability. So in this case, you have one complex that just serves, is biochemically identical, but otherwise is in a conformational and functional distinct state that just serves for the activation of this one. And this, as I said, occur in the context of chromatin. Once this has happened and several nucleosomes have been methylated, then the system obviously will switch to this state that is a lot more efficient, because now you can use 100% of the PRC2s for this modification to just do the classical spreading of the modification. Okay, so I told you the people that were doing the work just Vignesh was helped on the ubiquitin nucleus by these two talented Berkeley undergraduates that now are doing their PhD, and Trinity is working on the inhibiting modification that I didn't tell you. Now, just want to say, does the streptavidin monolayer limit the resolution that you can get by cryo-EM? And I want to show you that this is not the case with another study that was initiated by Basil when he was a postdoc in my lab, actually working on TF2H. Now he has his own lab at the Institute of Cancer Research in London. And this is a study, picobillisome, which are light antenna. This is work that was a collaboration with Cheryl Kerfield, who's an expert in this area, and her Spanish postdoc, Tina, who's now back in Spain and has her own lab. So these are structures that actually channel the energy of light all the way to the photosystem in the membrane. And the problem when Basil started looking at it was that he tended to have preferential orientation, but also the disc that formed this rock tended to fall off by effect of the air-water interface. So Paul, who was working on the PRC2, decided when Basil left to take over this project and try with the streptavidin monolayer. In this case, he biotinylated, aiming for about 1 biotin per complex, using this reagent, which actually allow you to quantitate this. So this would happen on a random lysine on the surface of the complex, which is, by the way, about 6 megadalton in size. And he would bind it to the streptavidin monolayer, and the benefit was that he could concentrate the sample, get random orientation, and better preserve. And it really worked very beautifully. And he was able to get a structure at an average of 2.6 angstrom resolution, where he was able, with the team from Cheryl, to fit the chain of 320 proteins and almost 400 billion chromophores. But we had actually reached Nyquist when we got to that resolution. So we actually prepared sample and we took them to Eindhoven, where we worked with Abai, and used, apart from anything else, a smaller pixel size, but also took advantage of the more stable cold field emission gun and the new filter system and so forth. And we have been able to push the resolution for these core elements from 2.6 to 2.2, for the rods from 2.1 to 1.9. But furthermore, when we concentrate on the central disk and impose symmetry, we've been able to now reach 1.6 angstrom resolution, and the animation will not move either. So you will not see the fitting and the fitting of the waters and things like that in there, but hopefully you can see the resolution. The thing that is very interesting is that we've used this method to actually start seeing hydrogens. So we don't see all hydrogens, but we see some. You can take the model that lacks hydrogen and do an F4 minus FC and look at the difference. And we see them where they're expected to be seen at this resolution that are, like, for example, along the hydrogen bonding in alpha helices. So obviously, the streptavidin monolayer is not a problem for obtaining resolution. If anything, it helps because it's preserving your sample. And this is just the last thing that I wanted to tell you. This is actually very cool from the point of view of morale in the lab. Imagine for Paul, he's stuck at about 8 or 9 angstrom for PRC2. He's gone better now with some latest software releases. So it's more like 6, but it still is not 1.6. But also from the point of view of modeling the energy transfer, just having that accuracy makes a huge difference, the modelers tell us. So it really was worthwhile collecting this additional data in this particular case and doing this study. And with that, I'll finish. I'll thank you, and hopefully you'll have some questions. Thanks so much. I have no idea what none of the animations played. It never happened to me before, but hopefully it didn't matter.
That's a lot. So questions?
Yeah, I want to make a comment about your comment of the limitation. I could not agree more with the fact that right now the limitation, the whole workflow is verification.
Yeah.
And I know I've learned the hard way. So, but your trick with it, which is very good, doesn't work with something very close. So we need somehow to find a way for a more universal way.
Right. So in principle, the spectacular monolith could be fairly universal. Yes. The problem is you have more steps. You have to biogenerate the sample. You have to create the spectacular crystal, which is the less reproducible part. You cannot do normal blotting individual. It has to be one-side blotting. It is harder to get the right ice thickness under those conditions. And then you have the additional step of removing the crystal lattice. So this is something that if you don't have to, you don't do it. If you can just solve the problem with using the right detergent, for example, you do it, or the right cross-linking. Well, the cross-linking is not compatible with where no detergents have solved your problem. This is the most universal thing that we have tried. It's just many steps. Every time you add a step, it could be a possible problem.
Now, talking about the cross-linkers, because this technique normally work with lysis. There are others who can be used with other residues. So have you tried any of this?
Yeah. So maybe we should try it. We have tried with cross-linkers that use aspartates and glutamates, but we didn't succeed to start with, and we've moved on. I think now that we've been hearing about alternatives, and it may be worthwhile trying again. It's just for PRC to really kind of nail it. We just have a system that works very well, but still, if we did the cross-linking and things look as good as what is looking now, and we could do double blotting, not having to worry about getting consistent as having monolayers, we will, even in that case, we will move.
Any other questions here in the room?
In addition to the methodological problem related to climate procedure, I also can imagine the biological problems of this analysis. Something that you have, for example, to miss the present that you were using one PCR2 molecule and two nutrition. Probably you have tried many other approaches. Can you, this is some kind of a magic, but you have to try many.
You mean in terms possibilities of preservation?
Structure complex.
Right. I mean, we could, we could use different chromatin templates. Yes, absolutely. So biological implications for being able to look at more sample, more complex without having to start all over again. The fact that, imagine how incredibly efficient, because it will take much less microscope time, much less computational time. Now we spend all the computational time getting rid of bad particles. Is the truth? It's all of these, chain of 3D classification, just throwing things away. And then there is so efficiency that actually is less money, much less money. But it's also biologically, to imagine right now, we can say, there are 3 different conformations that we can detect. And you would say, in principle, they reflect an equilibrium. But that will be true when we are just ultimately using 5 or 10% of the particles. That will be true of the equilibrium in solution only if all the states are equally affected by the air-water interface. But this is not, we cannot assume this. I mean, it could be that the air-water interface is affecting 2 or 3 states and not other. It could be making completely disappear a certain state. So if we wanted to get quantitative about defining the equilibrium of a mixture, we cannot be throwing away 90% of the particles, period. Because then what is this that you're describing? Yeah, those particles exist, but the proportion and whether they are the only ones, even if the software was fantastic at describing what you have at the end maintained, all the particles that you have thrown away because they are non-consistent due to different levels of damage. It's over.
I think we have some questions from people.
No, but there's also, if anybody could want to comment about the part that have to do with women and Nobel Prizes or not, any of that, I would love to hear your ideas of whether I'm delusional or making things up, or this is something that you guys have experienced or that makes sense or anything like that.
So you said that there were 3 things.
No, they were 3 things out of many.
Yeah.
Okay, but 3 that I brought up. Yeah.
The third one, that we are against ourselves. I think partly that's to do with society and how we have.
Right. Yeah. Self-perception.
I need to repress that.
Right.
Basically.
We have unconscious bias against ourselves.
To become aware of it and to call it out and to just push amongst ourselves.
Yeah.
Push ourselves to.
You know, I just want to, the Americans have this expression, if God give you lemons, make lemonade. You know, I think that being super self-critical is actually a virtue for a scientist. It means you double check, you know, I think it's great until it paralyzes you. If it makes you be critical, it's really good, rather than, you know, the first result that you get is, oh, I'm so good, look at what I've done. Where you say, okay, let's see, let's double check. Like, this is great. Rather than making you more critical, it paralyzes you or it makes you lose confidence or makes you not promote yourself. That's a negative. So just make the best of it and celebrate your self-criticism, but make it a constructive criticism. It's easier said than done, but that's why, you know, just talking to other people, and, you know, I was next to Jennifer when she was going through crisis, and like, really, you. So it all happens, but she uses it very, very positively. She just gets over it and just keeps going, and, you know.
Okay. Anything else? Not this time, Eva.
All right.
A mí me pasa lo mismo con las películas, tienes que mirarlo cada vez, porque de vez en cuando, por razones que desconozco, las películas dejan de funcionar. Entonces yo lo pruebo y si no funciona, me la vuelvo a cargar. Pero, o sea, me pasa continuamente.
Hola, ¿qué tal? ¿Cómo andas?
Un poco justo.
Bueno, no pasa nada. Al principio